Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing another really amazing guest today involved in creating a better tomorrow for so many people out there. Today, we have the honor of being joined by Dr. Joni Rutter, who is the director of the National Center for Advancing Translational Sciences, also known as NCATS, at the United States National Institutes of Health, where she oversees the planning and execution of their center's really complex, multifaceted programs that ultimately aim to overcome the scientific and operational barriers that impede the development and delivery of novel therapeutics and other health solutions. Prior to this current role, Dr. Rutter served as deputy director of NCATS, involved in collaborating with various colleagues across government, academia, industry, various nonprofit patient organizations, ultimately to establish the interactions and the appropriate collaborations with the NCATS programs. Prior to NCATS, Dr. Rutter served as the director of scientific programs with the NIH's All of Us research program. They're leading this really interesting scientific development implementation of their gathering of over 1 million United States participants to help advance precision medicine programs. And also at a time at NIH, she led the division of neuroscience and behavior at the National Institute on Drug Abuse. She is extensively published, is involved in speaking engagements around the world, many awards. She received her Ph.D. From the Department of Pharmacology and Toxicology at Dartmouth Medical School, went on to the NCI in the Division of Cancer Epidemiology and Genetics to do postdoc work. We're honored to have her with us today. A lot of really interesting and important themes to get into today. But Dr. Joni Rutter, thank you so much for taking the time out of your schedule to come talk to us for a little while.
Hey, you bet. Thank you so much for having me. I'm excited to have the conversation.
Yeah, I've been really looking forward to this. Joni, as we typically do, I'd love to turn things over to you at the beginning, just to talk a little bit more about yourself, a little bit about your background, obviously the development of your interest in pharmacology and toxicology genetics, and if you can maybe take us up to your dissertation, because I personally, I read through a little bit of cell type specific expression of MMP1. And I found in your dissertation sort of a little microcosm of everything you do now, right? I mean, here's a really interesting gene set that if there's too much of it, autoimmune diseases and cancer, too little, and we got wound healing problems. It's involved in rare diseases. Sort of everything that you've been doing along the way. Talk a little bit about how you got focused in that particular space as well, if you would.
Yeah, it's a big question. And thank you for asking it. I think you and I are probably the two people on earth who read that dissertation, except for my dissertation advisors. They did a really good job with that too. But I think just going back even before then, I'm originally from Kansas, a little town called Garden City, Kansas. My mother's name is Dorothy, and
Nice.
being from Kansas and with a name of Dorothy has sort of really set the stage for really thinking about things that are a little bit fantastical in a way. And being from Kansas, I was never really exposed to research, but during high school, it was my senior year of high school, and I had appendicitis.
Mm.
And it turns out that I had to have my appendix removed. And that was the moment, I think, the most salient moment, because for whatever reason, I don't know why this happened, but the doctors brought my appendix that they had removed to me in my hospital room, and they showed it to me, and they said, this is what we took out of you. And I thought, wow, that's really fascinating. I thought I'd be kind of grossed out by that, but it fascinated me, about surgeries, about medicine, about biology. And so that really sort of started my stage of going to college and being really focused on biology and chemistry. And then I spent about 3 years after college, before graduate school, kind of trying to think about what I want to do with my life. And at the time, I was really thinking about medical school, but I worked in this lab in Boston, and under Jim Gasella, and he was studying Huntington's disease, neurofibromatosis, a variety of rare diseases. And so it kind of recaptured that sort of research bug that I felt when I had that surgery. So these sort of were two events that culminated in my real interest in learning more. And so during that time of sort of between high school and graduation, high school and college, and then graduate school, I decided that it was going to be important for me in graduate school to understand a little bit more about genetics, understand a little bit more about pharmacology and toxicology. And those two areas at the time were really pretty distinct. In fact, we were just learning about genetics, and it wasn't as big as it is today, very early in the time of human genetics, for example. But I became very interested in that. So I went to Dartmouth Medical School for my graduate studies, and my project was on studying this gene that you talked about, MMP1, and its role in sort of wound healing, tissue resorption, sort of restructuring cells, cell-to-cell communication, all sorts of activities. So it was a fascinating enzyme. And it is also this collagenase 1, or MMP1, it's the same name. It's involved in essentially degrading the most abundant protein in the human body, and that's collagen. Yep. And it has been largely associated with how people get rheumatoid arthritis, and right. Because essentially that MMP1 enzyme that gets overactivated, it starts to degrade the tissue in the synovial joints. Mm-hmm. And when it does that, then you end up with very little space in between your bones, and they end up kind of connecting bone on bone. That's pretty painful. And there's a lot of inflammation and that sort of thing. And it just exacerbates then the expression of MMP1. So that's what was known going in when I started my graduate studies. But what I wanted to ask is, if that happens, then what's the milieu of the cell that makes it so but works so hard in rheumatoid arthritis? And it turns out that inflammation properties were associated with that. So interleukin 1 beta, yep. were cytokines that really kind of fueled how that enzyme was expressed. And then when I started to research it a little bit more, I wanted to understand, number one, what are the cells that do that? These were cells that were largely involved in inflammation. And then number two, I wanted to understand, well, gosh, MMP1 degrades collagen, and collagen's important for a lot of things. Maybe it's involved in other things besides rheumatoid arthritis. And so we started to look at cancer and how cancer metastasizes. Mm-hmm. And for the most part, the field had been looking at gelatinases, which are other matrix metalloproteases, or MMPs. And that was really the impetus for the work in cancer. But we realized that for cells to get into blood vessels, they have to break down the structure of the blood vessels. They have to intravasate into the blood vessels and then go in through an extravasation process. Mm-hmm. And through that, most of it was thought to be through the basal layers with the gelatinase MMP enzymes. But the collagenase MMP enzymes were a little bit in the stromal areas of that section. And so it turns out that it's pretty important there as well. But on top of that, I found a polymorphism, a variation within the promoter region of MMP that seemed to kind of put on the gas for those enzymes to be expressed, and that seemed to be associated with risk for these bad effects of that sort of metastasis activity. So that was the work that I did in graduate school, and I don't get to talk about that very much. So thanks for bringing that up.
No, I think it's, I mean, aside from just being a cool theme, again, as we get further in our discussion and talk about translation and all the little hiccups along the way, it's just a nice example of how, our old thinking of like, here's a gene that does this one thing. No, there's a lot more intricacy there. So I thought it was, again, a very interesting place to begin, that you were looking at this stuff way back in the day. And I thought that quite fascinating. Again, as we look, as we go into PubMed, following your PhD, as mentioned in a bio, you head to NCI, you're working in cancer epidemiology, genetics, epigenetics, papers on, again, MMP and melanoma. You're working on BRCA1 and 2 in ovarian cancer, HER2 and breast cancer. And then in 2006, something else pops up on sort of the PubMed radar. And I think this is your first paper, and I guess what you describe as neuroepigenomics. The title of this paper is Symbiotic Relationship of Pharmacogenetics and Drugs of Abuse. Another, obviously, like cancer, like the autoimmune diseases, major unmet medical need, and we'll be talking more about it. What got you looking at drugs of abuse alongside your oncology work at the time, and obviously your segue over to NIDA?
Yeah. When I hear these things, it does sound a little bit disjointed, but I can assure you it was very deliberate. It's what was fascinating at the time is that we started to hear exactly what you're talking about, more about how genetics was being, gene to disease, and we also then started to realize that the environment plays a pretty big role too. And so what is the real intersection between genes and environment? Mm-hmm. And there's a great quote from James Ewing about 100 years ago now, gosh. He was the founder of the American Cancer Society. And he proposed that heredity is likely to contribute to the etiology of cancer through the indirect transmission of enhanced liability. So cancer might be I guess partly caused by making people more susceptible to it. And so this was sort of starting to get at that idea. Now I learned that lesson really during my postdoc studying HER2 and P16 and all sorts of other cancers, gynecological cancers as well, through BRCA1, and learning that actually women who had surgeries like hysterectomies or oophorectomies had a reduced risk for BRCA1 related cancers if they were positive for BRCA1. So what was that in that gene environment interaction? Just that surgery was really something that reduced the risk. And there were other factors that were associated with it as well, like how many children they had, et cetera. So all of these things, made me think, well, what is the environment doing to help us, or not help us, but that may be contributing to the susceptibility. So then moving to the National Institute on Drug Abuse, the reason I found that interesting was because really we thought as drug abuse as somewhat of a moral failing, back then. We don't think that anymore. Thank goodness. It's a biological problem. And now we're starting to really address it in that way. And I think it's even now it's long overdue, but I think that's been really a big game changer to the thinking about it. And that's largely a nod to Dr. Nora Volkoff, who's the director of that institute. Mm-hmm. But really the idea is that even things like smoking and cancer, we know that they're inextricably linked, but why is that? And what we found through the course of our genetic studies in studying people who smoke is that they have some polymorphisms in the nicotinic receptor subunit genes.
Yep.
And depending on which subunit is affected, and we found a very particular one that's expressed in certain regions of the brain that tend to control your addictive behavior, or if you smoke, you might continue to smoke if you have this particular gene, because it's getting activated by the fact that you smoke. So to understand really more about the relationship between environment and the genetics, you had to be exposed to that insult, or smoking, for example. You had to take up smoking at one point. But once you did that, then there was this cycle, if you had these polymorphisms, that would sort of increase your susceptibility to it, sort of going back to James Ewing's hypothesis there. And then we started to learn that, gosh, if that's what's exacerbating your smoking and you're becoming addicted, that gives you that susceptibility then to perhaps other kinds of cancers. It's not necessarily that straight line that this causes this, but when you start to understand that there are very different factors involved in very complex biology and disease, that gets you one step closer then to being able to understand how to start to address that.
Mm-hmm.
And the great thing about gene environment interactions is that if there is any way that you can think about mitigating the environmental risk, reducing policies around smoking, for example, then you're kind of halfway there. So those were the kinds of, I think, bigger conceptual lessons that I really was wanting to look into a little bit more when I was at NIDA, and thinking about how genetics really impacts that interaction of addictive behaviors, and then exacerbating some of that severity of addiction.
Mm-hmm. Mm-hmm. And then taking that triad of genetics, environment, and lifestyle, and sort of this systems thinking, one of the places that we need to go before we get to NCATS is, of course, your scientific leadership at the All of Us research program. And, obviously, it's great to understand those connections, but hey, if we get 1 million people or 2 million, whatever the case may be, and we can really have that database set to explore this much further. And we profiled Rachel Romani a couple of years ago at the Veterans Administration on sort of the sister program there in terms of the Million Veterans program. I talked to Chris Lunn actually a couple of years ago as well, sort of when things were more from the tech side. But take us through a little bit about what you learned and some of, I guess, maybe what excited you as you were standing up that science behind a very extensive program to bring so many data sets together.
Yeah, what excited me was, well, when I was at NIDA, I since I was a geneticist, I worked very closely with other groups across the NIH, NHGRI, the Genome Institute, National Library of Medicine, and the archiving of genome sequences for then further research. And so I was involved in quite a number of activities around developing policies, how to implement those policies behind genomics, and how to do that very responsibly in a way that preserves privacy for individuals, et cetera. And so I was involved in a lot of activities because of my role at NIDA around understanding genetics and environment, and also working with vulnerable populations through people who are addicted to medications or addicted to drugs of abuse. We wanted to make sure that those individuals were protected as much as possible. And so when I learned about the Precision Medicine Initiative, which is what it was called, very early on, it didn't really roll off the tongue. Precision Medicine Initiative Cohort Program was the name. So we went through a variety of ideas to rename it, and All of Us, the All of Us research program was the landing spot. And the idea there is I think very apt, because it really is about observing as many people as we can. 1 million people is the target. And if we can get a minority of the population, that's really, I think, what would really contribute a great deal to our understanding of how genetics impacts disease, because we would see a lot more of that genetic diversity if we have more diverse people who were enrolling in the study. And so when I came to the All of Us research program, I had a very sort of one-track mind in how to put the emphasis on populations who were underserved in biomedical research. And when you looked in the literature on genetics and population-based studies, you saw those studies that were really happening in white people. And they weren't really reflective of the broader diversity of our population. And so this was something that the All of Us research program set out to address. And so I was hyper-focused on really that idea. How do we get there? How do we make sure that we are enrolling a majority minority? And to do that, we did a sort of a broad undertaking on, who is underrepresented in biomedical research. It's not just the racial, ethnic diversity that we have. It's that, very much so. Mm-hmm. But it's also people who don't have access to care. It's people who are even children or older adults or the elderly, people who are living below the poverty line, people who have cognitive deficits, people who have different levels of education, intersex or sex and gender differences or minorities, and people who live in rural populations versus urban populations. We wanted to make sure that all of those different groups, relating to zip codes, we know how zip codes play a role in our health. So we wanted to make sure that we could address and encourage and engage those types of populations in health-related research. And so our goal was to have a cohort that was about 75% of all of these individuals who were underrepresented in biomedical research, and then 50% would be racial ethnic minorities. And so that's why we sort of called it the majority minority populations reflected in the All of Us research program. So that in and of itself, I think, was critical to setting up the program and engaging communities, engaging people who hadn't been engaged before in research, to bring them in, and then to really understand observationally, what are the health effects that they're seeing, what are the diseases that they have, what leads up to those diseases. Those are the possibilities that we have now to go back into that cohort and start to examine. So I think we're going to see really probably in the next 5 to 10 years, a lot more research coming out of that based on what's been there to date.
Mm-hmm. Mm-hmm. Excellent. So now we arrive at NCATS, National Center for Advancing Translational Sciences. And so everyone listening and watching, you can go to the website. There is a 2024 to 2029 strategic plan on sort of everything, a quite extensive document. And the document sort of starts off with a definition of translation, which is defined as process of turning observations in laboratory, clinic, and community into interventions that will improve the health of individuals and the public. And I think it's quite interesting, because I think most of us maybe erroneously sort of think of translation as that area, it's sort of defined as the valley of death. And we've done a lot of this great research, but because of funding issue, whatever, we just can't get across to that first clinical experience, and we're lost. But no, you sort of define translation much broader, as you were explaining. It's about understanding these polymorphisms here in the lab. It's about selecting the right patients in the clinic. It's about follow-up once there's product out there. Take us on a little walk through sort of your history of NCATS, and a little bit about how you think of translation. And obviously there's an extensive list of principles that guide what you're doing in the 2024-2029 document. Walk us through a little of that, if you would, Joni.
Yeah, sure. Thank you. You're right. That area of translation. So translation is the area between basic science and clinical science, and a sort of clinical application, really. And that sort of translation is all the stuff that has to happen to move a basic science finding into clinical applicability. And sometimes we have findings out of clinical research that kind of feed back into that loop. So it's not necessarily linear, this whole linear thing. This is the second time I've said linear, but it's not linear. it's really about that sort of bridge phase. And yes, so people call it Valley of Death. I want to start to elevate that to become the bridge,
Nice.
then the Valley of Death. But because typically the Valley of Death is associated with this idea of the preclinical work. So you find a basic science target or gene that you think would make a good target for a particular drug. You go to find the drug, and then you do all the preclinical work. Does it really hit the right target? Can you get it into cells? All of those sorts of issues in the preclinical space tend to be where we have that, you start to hit the Valley of Death, so to speak, in that more traditional sense. But as compounds move further along that translational science pipeline, from assay development to lead candidate to lead optimization of that candidate, you find something that's working really well after a variety of medicinal chemistry changes to a particular molecule that will enhance its binding potential or efficacy or safety or what that might look like. Mm-hmm. Those are the kinds of processes that happen here, and so there's a lot of areas along the way where that Valley of Death is actually very real, and so those projects tend to kind of die in that space. But that Valley of Death actually does extend through all the way through from that preclinical pipeline to phase 1, phase 2 studies. In fact, 90% of the time, things fail in phase 2 clinical studies. 90% of the time things fail, and about 60% of the time it's that phase 2, and about 25-30% of the time it's in that phase 1 phase. So safety and efficacy in going into first in human trials is a pretty big contributor to when those things fail. And so that is the quintessential sort of Valley of Death, and things get dropped at that stage. Our goal is to build bridges, to understand really what are the hurdles that we see along the way in that translational science pipeline, and use the science of science to understand what are the operational barriers, what are the scientific barriers, what are the financial barriers. There are a lot of those. And oftentimes what is an issue is not always scientific. It is more about the operational aspects. And so what we try to do is in translational science, we talk about the idea of that translational science is really addressing those scientific and operational barriers so that we can overcome them and bring more treatments for all people more quickly. Mm-hmm. And so that's really the what we're trying to build. But recognizing there is a Valley of Death, that points us to places along that translational pipeline where there might be those barriers, and where we want to understand, can we build better tools? Can we build better assays? Can we use artificial intelligence to help us navigate this better? So what are the tools that we need? And then even more broadly, we think about it too as not thinking about it in a one disease kind of approach, but how can we address more than one disease in this process? And so building tools that won't just solve one problem, but will solve many problems. Mm-hmm. So that's really, I think, the idea behind what translational science is. And I think it's also a big area where it's important to understand. You mentioned those key characteristics of a translational science sort of theme. Mm-hmm. Thinking about unmet needs, thinking about generalizable solutions, thinking about working in teams. Team science is a really big key sort of concept for translational science, because you never know where ideas are going to come from that can help us address a particular problem. Mm-hmm. And so that's, I think, a key area for us as well.
And as you innovate along both these paths, and when we'll talk preclinical, and then we'll separately talk clinical. I've watched some presentations you've given, and you've talked about technologies like induced pluripotent stem cells in terms of creating better assays. You've spoken about I guess related tool like organ on a chip that may help us maybe not entirely move away from this animal study or having to perform a certain phase 1 study, but really getting a better understanding of toxicogenomics, whatever the case may be, ahead of time before we get to the human. What are some of the, from the preclinical angle, what are some of the things that excite you, technologies that you want to see more of, whether it's in silico stuff, AI, and what are some of the things, frankly, you think are hype? And love to hear your perspective on the preclinical side.
Yeah, thank you. So I am probably I'm a forever optimist. So I like hype, but I also like to understand where the issues are with it, and adjust that. But sometimes I'm an early adopter by nature in a way. So it doesn't take long though now for me to figure out what is hype. And I think we learned a lot in terms of the AI sort of. There's a lot more hype early on than was really kind of probably needed. Less sort of rigor evidence-backed findings of using AI, but mainly because the AI didn't translate very well early on to the work in biology. And now, though, I think that we're starting to understand what's needed for transparency, for mitigating bias, and all sorts of things. So I think we're still a ways away from really seeing the true impact and value of that with high confidence, I guess I should say. But I think that where translational science is its best is really understanding where those opportunities lie and how to guide the field to really make sure we're addressing those key issues before they become barriers to those activities. And so I do think that there is hype out there around AI. And our goal is to understand how we use it responsibly and appropriately. And one of those areas, I think, is in drug development and how perhaps we could use AI-based approaches to think about, maybe what might make a compound more toxic. And can we identify those sorts of things in compounds before we even really get to putting some compound into humans? So that's one area. I guess what I'm really excited about, though, is this idea of, how do we address these bottlenecks? And you mentioned organoids and tissues on chips. And I think this is a big area, but it's still very early. There's a little bit of hype associated with it, too. It's still a little bit in the early phases, I would say, because we have done a lot of work in the area, but it's been very specific. So for example, this is one of those tissue chips. This is a lung on a chip. Mm-hmm. And so we've taken lung cells from individuals, and we've used induced pluripotent stem cells to regenerate the lung cells and put them on this particular chip. And what's great about this is and again, working in teams, we had to work with material sciences and physicists, etc. But this is a very flexible kind of material, because the lung, you have to breathe in and breathe out. And so you need that flexibility incorporated in your model. And so we can hook up vacuums to this to simulate that sort of breathing in and breathing out. And when we put cells on here, then we can put disease cells or healthy cells or both. We can add drugs to them to see how the drugs interact with the cells. What biomarkers can we find? So it lends itself to really a lot of new ways of pursuing research questions, and perhaps getting at this idea of this really high failure rate that I mentioned before, 90% failure rate in preclinical studies. And if we can figure out how to increase the predictability of preclinical information that leads into clinical trials, perhaps we can start to chip away at that 90% failure rate. And that's the hope, I think, for tissue chips and for organoids, etc. But now, so I showed you a lung on a chip. This is a different kind of a model. This is a kidney on a chip, and it's got vasculature and kidney cells on it. So we can titrate in looking at both vasculature and kidney. And this is sort of a plug and play version where you stick this card into a machine and it really does all of the work. And then we've expanded now to multi-system organs on a chip. So that's what this is.
Cool.
Here on my
That's
head.
very cool.
So this is a liver, cardiac cells, skeletal muscle cells, and a cardiac microelectrode array for motor neuron assessments. So we can look at one drug, for example, one medication, one potential medication on a variety of systems now in this way. And so this is about the size, a little bit bigger than a credit card. And we're getting better, I think, at really understanding what are the needs here, how confident can we be in using these types of different in vitro models that more reflect the human condition. And in preclinical work, the gold standard is using mouse models or rodent models, for example, to do a lot of that preclinical work. And that could be the thing that's really contributing, or one of the bigger things that's contributing to our failure rate. So if we can think about how to bring in human-based cell models in this way, still, not using humans to do those testings, and maybe we can actually get smarter about what should move into clinical trials. And now we're working on the idea of generating these multi-organs on a chip for clinical trials. And so if you are a patient who needs to go through a phase 1 clinical trial, maybe we can take your cells and create these multi-organs on a chip and put the drug in the chip before it ever gets to you. And so that's where we're headed with this. We're still a ways away. There's a lot of regulatory work that needs to get done. So we're working very closely with the FDA, people like Dr. Bumpus, for example, and our teams, people in Arkansas, in the toxicological programs there at the FDA, and then here at White Oak at the FDA labs there with these similar kinds of approaches. But our labs are really in this sort of, how do we build this well? How do we make platform-based kinds of tools that can be applied to just about any disease? And then how can we study them from there? And in addition to the tissue chips, we are also conducting experiments to understand how we can build 3D biofabricated tissues. And one of the ones that we've been doing, we have pretty good tools now is in the area of skin. Biology is fascinating. It's very layered, it turns out. So you can think of, your dot matrix printer is essentially what this is, but it's cells. So you can print cells, and you can print the different layers of the skin cell, of the skin dermis, and get those different cell types layered in a basically in a plate, a 24 well plate or a 96 well plate, and then test different compounds to see if they're what they do to skin diseases like atopic dermatitis, for example. And so those are the kinds of examples that we're trying to do and build with these more human-based, physiologically relevant platform-designed approaches to really address this issue, of predictability for medications that might then move into humans.
Yeah, it makes me think. I mean, a couple careers ago, but thinking back to the few clinical development programs I was involved in, where we had that QT prolongation study going on simultaneously to the other study, and like that one would get the blood pressure would stay up until those results came in. It's like, can someone create a nice heart that we can just this study and so on. But yeah, no, it's fascinating. I completely root you on. I think it's amazing the degree of sophistication that, especially the multi-organ chip that you were just showing us, has gone. So I'm excited for you on that front. But thinking, moving from preclinical to clinical, as you were just explaining, obviously, a lot of intricate thinking has gone into some of what you've been proposing in these areas. And I've read again a couple of your papers recently. One which was just recent in Journal of Clinical Translational Science in May, just talking about how to assess a clinical trial site readiness. I mean, not every clinical location is set up to properly do a clinical trial. And then you also, a couple of years ago, you published this paper with all these cell and gene therapy luminaries on ultimately, again, this gets into some of the rare disease stuff, but when we have these smaller cohorts, can we design these phase 0 studies? Can we incorporate, as you're talking about sort of the racial and ethnic disparities in these clinical trials, get patient advocates involved in the process to appropriately, get the right population in the study early on. Talk a little bit about sort of what excites you in terms of the clinical side and some of these novel models that we may consider as we move into the future.
Yeah, this is a really great question, and it's one that we certainly haven't solved yet, but I think is super, super important to continue to build on the foundation of these new methodologies for clinical trials. And the impetus behind that paper on site readiness, for example, was the COVID pandemic. And early on in the COVID pandemic, we saw hundreds of different groups who were doing clinical trials, but on tens of people. And these were just not going to get to definitive answers. And we knew very quickly that we had a problem on our hands. And this is where the sort of the federal government and industry came together in a public-private partnership called ACTIVE, and accelerating COVID-19 therapeutic interventions and vaccines. Whew, that was a test. So that's ACTIVE. And the ACTIVE program just very quickly realized that this was a problem. And the reason it was a problem was because all of these smaller trials, not only were they not going to get definitive answers, but they were going to compete for the same patients who were coming down with COVID, that we wanted to make sure were in studies that could give us definitive answers. And so ACTIVE was a program that came together with a variety of NIH organizations. I did not lead it. We were a part of it. We were a part of something that was much bigger. And this is really a credit to just a number of people. But Francis Collins really led this effort from the NIH perspective. And there were a lot of industry heads who also led this effort from their perspective. But we came together, we said, okay, what are the key drugs or therapeutics that we should be trying on this infectious disease that affects the respiratory areas? And we come to find out a lot more than that. And so how do we think about a governance to do that? We need somebody at the real helm. That's Janet Woodcock, who back in the day was really the one there saying what we needed and how to get it done. And the beauty was we essentially had the ability to do a lot of those studies because of supplemental funding that allowed that to happen. And so we were able to pull together these groups and coordinate these efforts rather than compete with these efforts. And through that, I think we had a lot of lessons learned, like how to build a governance around these sorts of things, how to share data better, and all of those sorts of ideas were very important. And so for that site readiness trial, it's really building upon that. Okay, how do we make sure that our, for example, NCATS supports one of the largest programs at the NIH called the Clinical and Translational Science Award Program. We fund it's about 63 medical organizations across the country. And so what can we learn from how they've contributed to the pandemic and these clinical trials that were ongoing during the time of ACTIVE? What were the key features that made them ready to do the clinical trials? And so it's about having the right staff in place in the particular medical centers and locations that we're doing these studies, about understanding the biostatistics, expertise, patient engagement. All of those things were domains that are necessary for being site ready to be able to really conduct, I think, a definitive clinical trial. And that's what that paper was really meant to sort of gel together and create for a community of people who are doing clinical trials, whether they're from a large medical school or from a federally qualified health system or otherwise. We want to give them the resources to have them arm them with the ability to do sort of a self-check, a self-assessment of what their site readiness is, and then perhaps what they need to do in addition. Where do they lack in certain domain areas that they need to strengthen? And that's really the idea behind that paper, was to provide that bit of a roadmap and then strengthen basically our capacity and ability to do clinical trials like this moving forward, while really keeping what we used after the pandemic, or after the sort of the main emphasis on the clinical trials around the pandemic, is how do we keep things warm?
Right.
And to keep the clinical sites warm, this site readiness is meant to provide the tools for them to be able to do that. Yep. So that's really the impetus behind there, and I think it's been a critical addition to the literature, just to provide that as a resource for these different programs.
Let's talk a little bit about rare diseases now, because I know this is an area you're very passionate about, and here we have this estimated basket of somewhere between 7,000 and 10,000 rare disorders, 30 million patients in the United States, 300 plus million around the world. You published at the end of 2021 on sort of this ideas initiative showing that these diseases are expensive, the clinical development, the medical costs are expensive, and we need to again have innovation. NCATS has, on one hand, work going on in sort of the advancing gene editing technologies, and so sort of the cutting edge stuff that we hear about, the CRISPR tools and so forth. At the same time, I thought it was really fascinating. You were on a panel recently at the Milken Institute with David Feigenbaum down the street here at University of Pennsylvania in the Castleman's initiative. I had him on the show last year or so, talking about repurposing. Again, there's maybe many low technology approaches that we just, have put by the wayside. We got a lot of drugs out there that are doing X, they may do Y and Z. Talk about how you balance some of this, especially, as you look at this extremely important basket of diseases that is truly the unmet of the unmet conditions.
Yeah, that's exactly right. There are definitely unmet needs, and quintessentially NCATSian, I think. So just to take a step back, even on the rare disease side, I am indeed passionate about rare diseases. My mother had a rare disease, and so my story about my mother named Dorothy is a little bit different. And she had a rare disease, and it took her about 15 years to get diagnosed. And then once that she was diagnosed, there weren't any treatments, but there was a clinical trial at the Mayo Clinic. And being from Kansas, that's a pretty long distance to travel for a clinical trial. She was an older woman and would have had to do that mostly kind of by herself. And that was not a price she was willing or really ready to pay. And so we navigated that. So understanding sort of what those translational science hurdles are, these are those operational ones. Being from Kansas, didn't have really someone in the area who knew a lot about the disease. And then when she was diagnosed with it, treatments weren't necessarily available, and even access to clinical trials was very challenging. So these are those kinds of operational bottlenecks that we're also trying to address in the rare disease space. Things like telehealth, telemedicine, those sorts of things I think can really help rural communities, but there's a lot more that we need to do. And as you point out, the issue of rare diseases, 30 million people in the US, 350 million, 400 people worldwide with a rare disease. And the economic burden that you highlighted, I want to put a number there. The economic burden is about $400 million per year in medical costs alone for a person with rare diseases. So we did a lengthy study to really examine this by going into medical records. And then once we found an individual with a certain rare disease, we went back in time in their medical records to understand that the time and cost before they were ever diagnosed. And adding that all up, we came up to about $400 million per year. Now, at the same time, there was a group called the Every Life Foundation for Rare Diseases that did a very similar study. Theirs was actually much deeper. They looked at a lot more rare diseases. They came up with a very similar number of that $450 million of medical care costs for people with rare diseases per year. But what they did was super, I think, valuable. They didn't just look at the medical costs. They looked at the costs to the family, to what kinds of changes they needed to make to their home, to missing work, all of those sorts of things that come into play when you're taking care of a person with rare disease in the family. And so that total economic burden on the rare disease family or patient is about $1 trillion in medical costs and indirect costs per year. That's a lot of money. That's bigger than diabetes, bigger than Alzheimer's disease. So this is a public health problem. So despite the fact that rare diseases are rare, the enormous cost and economic burden to society, it tells us that this is something that we need to address. Not only that, if you take sort of the idea that as a geneticist, one of the really early kind of big wins, I think, in genetics was looking at families with hypercholesterolemia.
Yeah. And
by looking at families with hypercholesterolemia, this was a very rare kind of disease in families. But what that did by studying those families with rare disease, is it helped us get to the idea of statins. And now we have statins that are just very broadly used and have saved countless lives, probably tens of millions of lives, because of the impact that those statins have done in terms of the heart health and vascular health, et cetera. So I think the idea or the concept that studying rare diseases isn't just about the rare disease itself. It's actually can impact common diseases as well as other diseases. So This is why I get really excited about talking about rare diseases, because even though they are rare for any individual rare disease, the impact of understanding and perhaps even finding a cure for them can be very broadly felt. And so that's my motivation there. And also, as a geneticist, one of the things that is really exciting me is that I spent my whole career essentially looking at these different polymorphisms and how they might affect disease. And we use this term called association because it wasn't a correlation necessarily. There were so many factors that go into what causes a disease and then what makes one very severe. And so thinking about a polymorphism of a particular person has that might be associated with disease, and thinking about, well, should we develop a therapy for that? Probably not. We just don't know enough at the time from just looking at polymorphisms. But the tools and technologies that were created over the course of that, last 30, 40 years in this space, restriction enzymes, Taq polymerases, DNA ligases, nicases, all of these things that we used in the molecular biology lab back in the day, that led us to really where we are now and being able to discover the usefulness of Cas and CRISPR, Cas9 approaches for then thinking about really these gene-targeted therapy kinds of approaches. And so here's where I think all of the work that we've done in those single nucleotide polymorphisms and their disease associations, it hasn't just deepened our understanding of human heterogeneity, but it's also really kind of paved the way for personalized medicine and targeted therapies that now we're able to sort of enhance treatment effectiveness and maybe even reduce adverse effects for medications. But in terms of the precision medicine piece, that's what I wanted to kind of highlight for you too. Thinking about how we can then translate this into sort of genetic therapies. Right. These are sort of therapies that might be definitive, cures even. And we've seen some of this already start to happen with Duchenne muscular dystrophy, for example, spinal muscular atrophy. These are some of the ones that have come out, and hemophilia. These are areas that are these are the leaders in this space, and I hope we'll expand really much more broadly. And here again, I think is where our NCATSian kind of an approach is needed. When you think about rare diseases, and there are over 30 million of them, 80% of the time, those rare diseases are caused by single gene mutations or single gene disorders.
Sure.
So the bulk of rare diseases are caused by these single gene disorders. So if we can figure out not just treating the disease, but treating the gene, how do we go about treating the gene? So that concept, going from let's not talk about the disease, let's talk about the gene, get to the root cause of the problem. How do we fix the gene? Well, 30 years ago, that seemed nearly impossible, but because of all these tools that we've had along the way, and CRISPR-Cas9, and now we have base editing approaches, prime editing approaches. These are the ability to fix one single mutation within the genome, very specifically, very directly. And so the precision that's associated with that is truly spectacular. And here's where I think this idea of treating more than one disease at a time is so critical. But the bottleneck or the hurdle that is the challenge for translational science, I think, is how do we think about delivering these to individuals? So if you need to deliver a gene for a gene therapy, or you need to deliver the molecular tools to do a gene editing approach, or a CRISPR-Cas9 approach, we need to figure out how to deliver that effectively to the right cells at the right place. And that's not so straightforward. So our approach is really to think about that delivery side. And to do that, we want to understand sort of what is the biology for the viruses that we use to deliver for gene therapy. We use one of the main viruses we use is adeno-associated viruses, because these are relatively benign. It's weird to talk about viruses during the pandemic, but these viruses specifically are relatively benign, and they can hold, a good number of genes. They have a size limit, but they can hold a certain number of genes that fit that size. But they come in different flavors, and so they can also target very specific cell types. And so if you have a liver metabolism disease, or you have an eye disease, you would use two different kinds of adeno-associated viruses to deliver the gene. But what we want to understand is how can we build the viral capsids so that they can really be, we can do all of the studies in terms of toxicological effects, or how many of those curative genes get into the capsids. It's not 100% of the time. Sometimes it can be very low. And then how do we make sure that once the viruses get into the cells, that they're actually making the change that's needed, expressing the gene, for example. And also that's kind of a low fidelity. So we want to enhance all of our abilities in understanding the biology of sort of that viral gene interaction, and then putting that into a delivery approach for this. So that's one of the areas that we're really trying to study now. And we do this, we have a couple of different programs in this space, but one of them is the Bespoke Gene Therapy Consortium. And it's an accelerated medicines partnership with the foundation of the NIH. And it involves, we have about 30 partners all told, about almost a dozen NIH partners, and over 2 dozen industry partners, academia partners, and different patient organizations that are involved as well. And so we're all coming together to try to understand what these, how to really make this program work, and how to identify some of the early phase clinical trials that we can test this on. And we've just highlighted 8 different clinical diseases that we're going to be kicking off with this particular approach. And that will be the real test. We won't start these clinical trials for another couple of years, but we have, now we've identified them and can start to work on what's the viral flavor, the AAV virus that we want to work on. How do we enhance the biology for that? So now that we know the endpoint of what disease we want to work on, how can we then back translate that to build sort of a manufacturing pipeline to get us there better, faster, and more effectively and efficiently. And so that's why it's really important that the entire ecosystem comes together to be able to do this. And so that's the plan with this particular consortium. And then another sort of similar kind of program is through our Common Fund program at the NIH. This is a program that supports activities that can't be supported by any one particular institute or center at the NIH. Okay. But it needs all of us, and the work that gets done in that program can help all of us. Mm-hmm. And one of these particular programs is called the Somatic Cell Gene Editing Program. Okay. And so this is really focusing on the gene editing side. And so gene editing is really more about finding a particular mutation that you can use gene editing tools like CRISPR-Cas9, but more recently there are base editors and prime editors by David Liu in his lab. Mm-hmm. That have been, I think the fidelity of the editing is getting better and better with these new tools. And so using these tools, we're doing the studies in small animals and large animals before they go to humans to understand how we're able to make sure we're seeing the edits taking place, for example, in the cells that are meant to take place. And then hopefully we will graduate those into clinical trials in the next few years. So what's fascinating to me about this whole space is, I mentioned before, it's a 90% failure rate to get to the clinic, and it's $2.6 billion. It's even more now to do that. But also it takes 10 to 15 years to get to a clinical trial. And after that, it takes another 15 years to get into the medicine cabinet. So it takes a lot of time. But this field is moving incredibly fast. Yeah. And we just got these. We're just starting to hear more about how prime editing and base editing and CRISPR-Cas9 approaches can be used. And we're starting now to talk about clinical trials already. And that's because we have more tools that are very specific, very precise. And I think we have a big opportunity here to make a dent in this idea that with, 30 million people have a rare disease, but there are about 10,000 rare diseases that exist. So with that, only 5% of them have therapies. This gives us a really big hope that we can start to tackle that 5% number to get it up to a higher percentage. I'd like to see 25%. I think that's pretty high number. It's pretty audacious, but I'd like to see something like that. And I'd rather fail at 25% than succeed at 6%. So this is the idea that I think precision medicine, gene editing technologies, gene therapy, antisense oligonucleotides, all of these sort of tools now are really things that we have in our armamentarium. And so when you look at a rare disease and a specific mutation in that rare disease, you might say, okay, well, you need to replace the whole gene here. This is a gene therapy kind of treatment, or this is a single gene mutation where we can hit a muscle and it'll fix it right, or hit a cell and it'll fix it right there. Mm-hmm. And that will be sufficient. So we can start to look at the modality needed just by understanding a little bit about the gene and the disease. Yep. And so then you can start to think about this as a platform that's really almost like a Swiss Army knife for rare diseases. So where do we apply it? But we have the tools to apply that. It's just which tool do we need to use when? Those are the kinds of questions that I see in the future. So that's how we're trying to build that better tomorrow for today.
Awesome. Yeah, and it's again, it represents your strategic principle. I mean, it encapsulates collaboration and innovation, and obviously, generalizable. So it's awesome to see this happening at that level, especially with the 5% and where we need to be going with that. So I really applaud you on that one. And again, I was just wondering if we could go to one other place while we have you, Joni. I so appreciate your time today, but going from rare to decidedly very unrare, unfortunately, is this topic of opioid misuse and addiction. Obviously, this feeds all the way back to your original work in the area, but I don't know if therapeutics for ending addiction are rarer than I say maybe, or the pipeline is drier than maybe the antibiotic pipeline, but it's probably pretty dry, I would assume. But where are we? Just say a few words to you about the HEAL initiative, which stands for Helping to End Addiction Long Term, and just sort of how you look at opioid addiction and novelty there, because clearly I don't, I have friends in pharma. I don't know how many are involved in developing opioid, or reversal of addiction drugs nowadays. But where are we in 2023?
It's a good question, and it's not a good one. I mean, it's not a good story here.
Yeah.
But I think there is hope in a way. The Helping End Addiction Long-Term program, it has been an initiative that's been happening for the last 5 years at the NIH, and it has helped shine a light on this problem. And I think that over the course of the pandemic, especially, what we were seeing too was a very robust, raging epidemic of opioid addiction, and that has not really faltered. So this is one of those things that, again, requires the entirety of the ecosystem to come together. And this is a program that's largely been conducted through the Office of the Director at the NIH, but also under the leadership of Nora Volkoff and Walter Koroshetz at NINDS, the National Institute of Neurological and Stroke. Neurological diseases and stroke. Boy, acronyms today. So NINDS, and their impetus is really thinking about pain. They've studied pain for a long time. We still don't have a really good handle on what are the biomarkers of pain. How do we measure pain? This is a long-standing problem. But obviously, pain is intricately linked to the opioid addiction, because when we're in pain, this is really one of the few things that helps. And so when you think about morphine, sort of then it lends itself to moving into other things. And now fentanyl, those sorts of really strong drugs are just very powerful. And people who use them often don't recognize the real risk and dangers that they have. They're trying to figure out how to work with their pain. And then, of course, there are people who have these more the severity of addiction in the area of opioid addiction. And so HEAL is meant to try to understand that intersection of pain and opioid use issues. And I think that they're approaching this in a variety of different ways. There's a lot of preclinical activity going on. And, I would say that one of our key principles at NCATS is to take bold and rigorous approaches. And what that's, it's sort of a keyword or a euphemism for taking risk and essentially de-risking some of those areas. And pain and addiction are some of those areas that need a bit more de-risking, because you're right, there's a dearth of industry companies out there who are working on or in this space. And so how can we identify particular targets that might be sort of unique or different than the typical targets that we've seen, the new opioid receptors or the other opioid receptors that might be targets? Those maybe won't be the ones that we want to go after. What are some of the sodium channel targets that maybe we should be thinking about? So the idea is, how can we kind of open up the aperture of the targets out there that might be useful for pain and addiction, and that individually or at that intersection. So a lot of the work that's going on preclinically is to develop tools. And in fact, we have a tissue chip program, for example, in space to develop new models that we might be able to use. And so, for example, I talked about arthritis before. That is one of those areas that is very painful. And so people want to treat that. And that is an area where you get more addicted. And back pain, for example, diabetic neuropathy, for example, these are all areas of diseases that lead to these painful conditions. So understanding sort of how to build those models and then perhaps find these new targets for treatment, that's one of the areas I think that is a promising area for that research. And then on the other side, on the clinical side, for the translational work that we're doing there is really to look at perhaps how we're treating pain. Are there things that we can learn on what are the treatments that are used these days? How should they be given? How can we do comparative effectiveness studies to see what is going to be beneficial for certain populations of patients, for example, with different kinds of pain-related diseases. So those are the kinds of areas that we're working on in the HEAL program. And I think we've made a lot of progress overall in the entire program on developing new IND approved compounds for further use, and also just developing a cadre of ready clinical trials that can really accommodate hopefully the future transition into that space. So that's kind of what the HEAL program has built to date in kind of a nutshell. I didn't really do it a very good justice, but in a nutshell, that's really.
No, I'm just glad that somebody's thinking about it like you are, and it's clearly a very unmet area, like most of these are. But the fact that you're giving it attention and an extremely important time is just, again, I take my hat off to you for that one. Joni, I really appreciate your time. Anything while we have you that I missed? Any other upcoming initiatives, public facing stuff that you want to mention? Please take the floor as we begin to wrap up. Anything else, please?
Yeah, one area I will just touch upon. I usually try to weave this in, but there were so many great questions you had. I didn't get a chance to weave it in. But another area I think that's really important for translational science is the area of data science in general.
Yeah.
And we talked sort of initially about artificial intelligence, and this kind of gets back to that. But certainly machine learning, right, I think has been a big thing that we've been using. And we want at NCATS we through the course of all of the different kinds of studies that we do, we have a lot of data that's generated. And so an important aspect for us to be thinking about is how do we turn that data into knowledge? And so what are the tools that we need to be able to create that knowledge? So data science, informatics, machine learning, and addressing sort of the interoperability features of data and being able to sort of compare one data set to another. Oftentimes those data sets are created with very different formats or different features, different variables. And so they're apples and oranges. So we want to try to encourage and adopt ideas where they're more interoperable so that we can compare different types of data sets. And one of the big areas I think that we've made a ton of progress in this space at NCATS is through the National COVID Cohort Collaborative. Again, because of the pandemic and the urgency that was there, we needed to do basic science, translational science, clinical science, data science, all at the same time for this pandemic. And so one of the contributions that we had for that is to build an electronic health record database for COVID-19. And that was just wildly successful. And I think it exceeded all of our expectations. But the key in that was doing the big thing that was needed that nobody else had really done before. And that was creating electronic health records so that they could talk to one another. And right now in our country, we have, if you go to one hospital to see your doctor, you have a medical health record system. But you might go across the street to a very different doctor that uses a very different medical health record, and those don't talk. So what we've done is we don't really care what system you use. We've created a way that we can bring in the data and translate it so that it is based on one common language, one common model. And so with that, we've been able to really solve the problem of interoperability with electronic health records. So we did this for the pandemic, and we've had a lot of insights from this research. We have over 3,000 investigators in this database. We have a lot of publications that have come out related to COVID-19, related to long COVID, and understanding different symptoms that might be associated in the health records there, et cetera. We also looked at Paxlovid use and those sorts of things, how effectiveness of vaccines, et cetera. But I think what the big value here is that solving that problem of interoperability, here is an area where it can really broaden to a lot of other diseases, especially rare diseases. With rare diseases, not only are they rare, but in the electronic health records, they're very difficult to find in the electronic health records. They don't have the code classifications that are used in electronic health records to identify particular diseases. So there are some good things about that, and there's some bad things about that. But that is one of those areas where I think that as we start to evolve in this area of data science, electronic health record use for research, and real world data and evidence, this is an area where I think we're going to see blossom a lot more. And I hope that will have a lot of impact for the patients out there that we really want to try to help. This is our goal, after all, is to help the patients out there and finding those insights. So that would be one area that I would pick up on that we didn't quite get a chance to talk about more. So thanks for asking that question.
No, I thank you for bringing it up. But yeah, it's it rounds off again this quite elegant approach that you've put together there. And I just, again, I'm rooting you on and all the whole team, and encourage everyone listening and watching the show to take a look at the NCATS website at the NIH, to look at the strategic document. Joni, I just so appreciate the time you spent educating us on these themes and everything you're doing. Again, for everybody who is going to be listening to this particular episode of our show across the various podcast networks or watching on our YouTube channel, again, you've been spending time with Dr. Joni Rutter, Director of the National Center for Advancing Translational Sciences, also known as NCATS at the United States National Institutes of Health. Joni, I again want to thank you for taking the time out of your schedule to educate us. Obviously, thank you for everything you're doing for everyone here in the United States and around the world with these diseases. And we'd like to say on our show, thanks for helping to create a better tomorrow via what you do. Really an amazing story, and rooting you on.
Thank you so much. It's been a pleasure talking with you, and we can't do it without everyone else. And I really appreciate the conversation today. So thank you to you and your audience.
Great having you.